So I'm gonna just jump into my presentation. So thanks, Scott, and welcome everyone in this Q&A session. So it's such a privilege to be here and presenting some of the insights, but also my PhD research a little bit. So my name is Putra, and I am a PhD student at Johnson's lab. And then in this occasion, I'm gonna present a little bit of insight and my research about the plant and microbe mutualism, relationship, conflict, and resolution. So this is kind of a very classical examples between flower and the pollinator insect, which is the honeybee, uh, which is the example of mutualism. So it's a biological interaction between species wherein both these species benefit with each other. So unfortunately, I'm not gonna talk about the relationship or mutualism between flowers and the insect, but today I'm going to talk about the visible of the invisible, which is the plant and microbe mutualism. So I'm gonna just simply divide my presentation into the three different sections. So the first one is about relationship, the second one is about conflict, and the last one will be about a resolution. So first, let's jump into relationship. So this is a very fun fact. So when I was typing it last night, this is kind of a familiar hashtag. So relationship goals. So when I type it, and then it appeared that almost 2 million results in Google, less than 1 minute, I guess, less than maybe 10 seconds. If you have social media, and then if you type this hashtag, and then it will appear about 20.3 million posts in Instagram. So this is a very kind of like a trending topic or trending hashtag that might also apply in plant and microbe mutualisms. And what are the goals of this mutualism? So I'm going to introduce with you two different plant species. On the right hand side is grasses, and the left hand side is legumes. So the first, I'm gonna explain a little bit with grasses. So grasses have mutualistic symbiosis with root endophytic fungi. It's called arbuscular mycorrhizal fungi that lives inside the root system. So it's one of the most ancient relationship in the terrestrial ecosystem. So it started since 500 million years ago. And what is the most interesting part is that almost 80% of terrestrial plants are colonized by these AMF or arbuscular mycorrhizal fungi. And what are they doing? So these arbuscular mycorrhizal fungi transfer phosphorus. So it's quite a limiting nutrient in the soil to the host plant in exchange for carbon-based photoassimilates. So plants do photosynthesis, sort of like they cook food for themselves and then transfer it into the root system for their symbionts. And so that's the below ground part of the plant. And then the second root symbiont I'm going to talk about is the leaf endophytic fungi that live above ground of the plant. This is quite specific. So the genus name is Epichloi. And so what they're doing, so they do protect the plants by releasing toxin secondary metabolites. So to protect the host plants against herbivores such as insects or mammals that feed on the grasses. So they can produce these specific toxins to protect the host plant, also in exchange for carbon. And basically, these two different fungi, below ground and the above ground fungi, are largely studied by a colleague of mine, Jimena, but also a student of Scott. But today's talk, I'm going to focus on the left side of the plant, which is the legumes, and that have a symbiotic association with nitrogen fixing bacteria, or we call it as rhizobia. So inside these tiny little structures called root nodules. And so these bacteria essentially capture the atmospheric nitrogen and then convert the atmospheric nitrogen into available ammonia through the enzymatic process using nitrogenase, and also in exchange of carbon photoassimilates. So apparently, so this mutualism involves a cost, but also benefits. So it's not really quite cheap to convert this atmospheric nitrogen into available ammonia for the host plant. So the bacteria themselves, they have to synthesize this enzyme, this specific enzyme nitrogenase, in order to convert this inert atmospheric nitrogen for the host plants. But also in the plant perspective, the plants have to do photosynthesis, and then they have to transfer all the resources, carbon resources, for the rhizobia inside the nodules. So it involves costs and benefits, and this leads into the second part of my talk, which is about conflict between the two. So this is a nice review paper from New Phytologist, so Joel et al. He explained about essentially legumes and rhizobia, they poses an ongoing conflict over the time. So for example, if I'm gonna try to explain this curve, and this graph or this curve corresponds nicely with these phenotypic images in the panel C. So if you can see, if the legumes have a little bit of nodules, it's fine. I think it's okay. And then like more nodules, the second one is also fine. You can see the plants are getting bigger, so it means they are quite happy with more nodules. And then with the third one, if you see here, that's the tipping point, or like the equilibrium, the optimal point between the two. So it's like this amount of nodules are quite ideal for the host plant growth, because the plants can also maintain the resources for the rhizobia inside the root nodules, and as a result, you can see here the plants are looking nicely, healthily, and also they can get more flowers as well. But then if we go further into this spectrum, so number 4 and 5, so it's not really the goals of the relationships, because it's entering the zone of conflict, where the nodules are quite abundant there, but the plants cannot really maintain the resources for the nodules, for the rhizobia inside the nodules. So that, as you can see, the comparison is that the plants are getting smaller. So they're pretty upset because they can't really do photosynthesis so much over the time because of the limiting resources and energy, or ATP. And then if you can imagine the last one, which is the 5, the 5th image, you can see there are lots of nodules occupy all the root structures, and then how can the plants compensate all the resources that they have to transfer to the root nodules. So it's very terrible for the plants, so that we can learn that mutualisms. I think one of the students here was asking, mutualism is very context dependent, but also I would say mutualism is unstable because in nature, organisms, plants do not live in isolation, so they interact with each other, but also at the same time they have to face stresses from the environment. So it's a costly investment between the two, but can also lead to the conflict of interest. But the interesting part is that in the nodules, in the that distinct root structures, it's not only harbored by rhizobia, but also it's harbored by a different kind of rhizobia. So it means that rhizobia inside the root nodules, they do not live alone. So there are some other microbiomes or maybe bacteria or fungi or archaea that have quite a mysterious role at the moment. We don't really know what they're doing, but they can be there and they can also manipulate these mutualisms to some extent. So actually, it's not quite straightforward, so to say. So and then now we are entering into the final part of my talk, which is the resolution. And I'm gonna just a little bit talk about silicon and why all of a sudden we are talking about silicon. So I'm going to try to explain it to you that silicon is a beneficial nutrient. So essentially, when plants take up silicon from the soil and then they accumulate it, they deposit it, so the plants can better defend themselves against abiotic and biotic stresses such as herbivores, drought, heavy metal toxicity, but also with the other climate extremes. So we know that, oh, silicon is a beneficial nutrient. And we also just learned that along a continuum, these mutualisms can be starting from the extreme spectrum from the left side, and then another spectrum on the right side, which is from parasitic to mutualistic. But it's not really black and white. So it's not really parasitic or fully beneficial or mutualistic, but somehow they are in the middle. So it means that in the case of legume rhizobia mutualism, some rhizobia are less effective. So they can still contribute a little bit of fixed nitrogen for the host plant, but they don't really do that much. So that's why it creates dilemma between the two. So that's why this is where my PhD comes in. I'm going to try to better understand how silicon supplementation affects these mutualisms, whether we can sort of stirring this relationship to have a better outcomes eventually. So previous studies all showed that silicon supplementation had positive impacts on nodule function and growth, but also the nodule activity. And I also confirmed in one of my PhD chapters that silicon supplementation essentially increased this enzyme activity that I already mentioned earlier, which is nitrogenase. And then we can conclude that, oh, it is good. So that silicon somehow interact with this sort of nitrogenous activity inside the root nodules. But also, the most intriguing part is that silicon supplementation can also increase the nodule silicification, meaning that somehow nodules can actively take up this silicon. So on the left side, you can see the graph from the A panel. So you can see there the comparison between non-silicon plants and then silicon-treated plants. So the control plants without silicon is the light gray, and then the black bars are the plants treated with silicon. As you can see here, silicon supplementation significantly promoted nitrogenase in these three different genotypes of the legumes. And then the B panel shows that actually silicon also promoted silicification, so process silicon being deposited in the root nodules in these three genotypes. And then when we try to analyze these root structures called nodules at the scanning electron microscopy facility at our campus in Parramatta. And we can see here, so the first one is the outer layer of the nodules. As you can see there are green spots. So it indicates that silicon is there, is deposited there. And then when I did a cross-sectioning of the nodules, and you can see here at the bottom of the picture is that silicon is also deposited inside the internal layer of the nodules. So somehow it led us into a direction that probably nodules, which is the joint structure between the legumes and the rhizobia, they can actively take up a beneficial nutrient from the soil for their own benefits, or probably they can speed up or accelerate certain process or metabolisms inside the root nodules that may benefit the outcomes of this mutualism. So this is really fascinating, but because of the sake of time, I couldn't really explain lots of our nice findings to you. But as a take-home message, we know that legumes are very nutritious. They are important crops, not only for us, but also for animal feed. So that it is really crucial for us to better understand these mutualisms and sort of how can we stir these ineffective mutualisms by any means, by any methods. We don't really know. I don't think there is like one size fits all kind of approach. So we can try different things to improve the legume productivity. So not only for us, but also for the environment, because if we realize that if we can improve this relationship, then we can reduce the amount of synthetic nitrogen fertilizer for our crops so that we can just utilize this natural relationship that already exists in nature. So by the end of my talk, I'm going to acknowledge and thank so much all the Silicon Group, also my supervisor, Scott Johnson, but also funding bodies that made this research and my PhD possible.